Hi, I'm David, and this is the Biology Classroom. This is a paper discussion video, so get ready with this paper and let's go through the questions and answers together. 1A shows us the Krebs cycle. Oxaloacetate accepts the 2-carbon fragment of acetyl-CoA and forms citrates. There are 3 Qs turned into Rs in the cycle. This is following the number of NAD being reduced in the Krebs cycle. When the 5-carbon compound is converted to oxaloacetate, it loses 1 carbon. It is removed as carbon dioxide in a process called decarboxylation. T and U refer to another hydrogen carrier, FAD. There is only 1 FAD being reduced per cycle. B. Describe how ATP is produced from ADP in the Krebs cycle. In the Krebs cycle, ATP is produced directly by substrate-linked phosphorylation. It is also called substrate-level phosphorylation. It is the production of ATP from ADP by directly transferring a phosphate group from a phosphorylated compound. In this case, the phosphorylated compound and ADP are the substrate, and ATP is the product, hence the name substrate link. This ATP production method does not involve hydrogen or electron carriers such as NAD and FAD. An enzyme catalyzes the reaction. C. Describe and explain the features of ATP that make it suitable as the universal energy currency. Due to its structure, ATP is small and water-soluble. It can move around the cell easily from where it is produced to where it is used. Do not say that ATP can move around the body easily, as ATP does not usually move from cell to cell. When ATP loses a phosphate group, energy is released. The reaction is called hydrolysis, and it is catalyzed by an enzyme called ATPase. Be careful when stating the enzyme. Many students mistakenly say that ATP synthase is the enzyme that hydrolyzes ATP. This is a quick process which releases energy immediately in small packets. You can also get this mark by providing the value of the energy release. Releasing energy in small packets can reduce the wastage of unused energy, which is eventually lost as heat. ATP can be recycled or regenerated rapidly in cells. The rate of conversion between ADP and inorganic phosphate to ATP and vice versa is very high, which we describe as a high turnover rate. ATP links catabolism, the energy-yielding reactions, and anabolism, the energy-requiring reactions. This is because ATP is synthesized during catabolic reactions and used in anabolic reactions. Question 2 wants you to outline how bacteria become resistant to antibiotics. The arising of an antibiotic-resistant gene in a bacterium is due to random mutation. This leads to a variation in the bacteria population where some cells have resistance, but others do not. When they are exposed to an antibiotic, it acts as a selection pressure. A directional selection occurs where one of the extreme phenotypes, namely the resistant group, is being selected for, while the opposite is being selected against. The resistant bacteria with an allele that codes for antibiotic resistance are said to have a selective advantage. They survive and reproduce. The resistance allele is passed to the daughter cells. This passing of alleles from a parental cell to daughter cells is called vertical transmission. Horizontal transmission can also take place. It is when the allele is transferred from one organism to another, not from the parent to the offspring. In bacteria, it can happen by transduction, transformation, and conjugation. The transmissions cause the resistant allele frequency to increase over time, as more and more cells are now containing this allele. It is a fast evolution due to the short generation time. There is an increased chance of resistance if people do not finish the full course of antibiotics. This is because those bacteria with a greater resistance are not killed when we stop taking antibiotics halfway, and they will reproduce and increase the number of resistant strains. It can also happen when there is overuse of antibiotics. The more a selection pressure operates, the more likely a species will evolve to adapt to it. There is a mark for AVP. For example, you can give a detailed description of how a resistant allele can provide antibiotic resistance to a cell. Question 3 talks about gene expression. A1 shows the leg operon, and you are asked to outline its main feature. The leg operon has a promoter, which is the binding site of RNA polymerase. It has an operator, which is the binding site of the repressor protein. The binding controls the expression of the operon. There are 3 structural genes, leg Z, leg Y, and leg A. You can also get this mark by naming the proteins they code for. A2. Explain the role of leg I gene in the regulation of the leg operon. This gene controls the expression of structural genes. It codes for the repressor protein, and it is always expressed. This means that the repressor protein is being produced all the time. Because of this, the repressor protein is said to be a constitutive protein. The repressor will bind to the operator and block the promoter. This prevents RNA polymerase from binding to the promoter. As a result, the structural genes are not expressed as transcription does not occur. However, when allolactose binds to the repressor protein, it changes shape. It can no longer bind to the operator. This causes the promoter to be unblocked, and hence gene expression can occur. Transcription and translation take place. The proteins coded by the structural genes will then be synthesized. B. Suggest and explain why it is an advantage to a prokaryote to have a repressible operon. Repressible operons refers to an operon that is usually switched on, but the transcription of the gene can be repressed by the presence of a particular substance. This means the enzymes or proteins coded by the structural genes are being synthesized continuously. These enzymes or proteins are the essential ones which are needed for the cell almost all the time. The trap operon is an example of a repressible operon. When the end product of the enzymatic pathway is at a high concentration, product inhibition occurs. This ensures that the product is made continuously until it is no longer needed. In question 4, we have some information about SCID and the treatment options. A. Suggest and explain why it may be more appropriate to use enzyme replacement therapy to treat SCID instead of a bone marrow transplant. A healthy and suitable donor is needed for a bone marrow transplant. Finding such a donor may not be easy, making the procedure much more difficult than the enzyme replacement therapy. This treatment involves the injection of ADA to the patient. It has an immediate effect as the enzymes start catalyzing the reaction right after the treatment. Besides, it is easy to administer the treatment since it is by intramuscular injections. The bone marrow transplant is done by blood transfusion, which is more complicated. The enzyme is produced by genetically modified E. coli that receive the human ADA gene. This means that the enzyme is identical to the human enzyme, so it does not cause an immune response and have little chance of rejection. Lastly, bone marrow transplant is much more invasive to both the donor and the recipient due to the collection and the transfusion methods. There is a mark for AVP. For example, the production of ADA by recombinant DNA technology can be done on a large scale. The mass production will make it cheaper. B wants you to outline the procedure used for gene therapy treatment of a person with SCID. First, the patient is given a drug to increase the number of stem cells in the bone marrow. This will increase the number of cells being collected. A viral vector is genetically engineered to contain the normal and healthy allele. Then, bone marrow is removed from the patient. It contains stem cells. The stem cells will be mixed with the viral vector. When the virus infects the cells, they transfer the normal allele into them. Radiotherapy or drugs can be used to kill some stem cells in the bone marrow. This is to make space in the bone marrow before the transfusion. Now, the transduced stem cells can be infused into blood. When the transduced stem cells divide and specialize into lymphocytes, they can produce functional ADA. There is a mark for AVP. For example, you can name the virus used as the vector or provide some other details in the procedure. C. Suggest the social and ethical implications of gene therapy for SCID that need to be considered before the treatment is carried out. You should discuss both the positive and negative implications. Many students lose marks as they only talk about the negative side. Let's look at the positive implications. Stem cells can divide infinitely and form new cells that also contain the normal allele. So, it is a long-term treatment that leads to a better quality of life. With that said, there is no need for regular injections or treatments. These will save time and money for the patients and the healthcare system. More money is now available to the health system in the long term, and this can improve the facilities, drugs, and services. No donor is needed to obtain the stem cells anymore, making the treatment more available and easier than the stem cell transplant. However, due to the high cost of materials and labor involved in the procedure, it is expensive. The extraction of stem cells can be stressful for the patient too. Bone marrow cells are extracted through a needle inserted into a bone, typically a pelvic bone. There might be cultural and religious objections. In some beliefs, the modification of DNA is considered unnatural and against the creator of the world. Lastly, some viral vectors may cause cancer in the recipients. Some patients develop leukemia as retroviruses insert their genes into the host genome randomly. This results in the malfunctioning of a regulatory sequence of a patient's gene and leads to cancerous growth. Question 5 shows the gene that determine the body color patterns of a freshwater fish. A wants you to calculate the chi-square value to compare the observed and expected values of the 4 phenotypes. The calculated chi-square value is the sum of O minus E squared divided by E for all groups. So, you have to do the calculations for each row and then sum them up. B shows us the critical value at P equals to 0.05. By comparing critical and calculated values, we can determine if observed and expected values differ significantly. First, make a statement to compare the values. In our case, the calculated value is lower than the critical value. You can state the values in brackets. This means the observed numbers are not significantly different to the expected number at P equals to 0.05. In other words, the difference between them is negligible. Any differences are due to chance. Chance refers to the random events which cause the difference, such as counting error, sampling error, and a small sample size. In C, we have to complete a genetic cross. The genotypes of the individuals are the combination of the gametes from both parents. X and Y are the sex chromosomes that determine gender. XX is female, while XY is male. Those with at least a big R on the X chromosome are red, the rest are white. With at least one big B allele, black spots are formed. Homozygous recessive individuals have no spots. D. Explain why there are no white males with black spots in the F2 generation. The Y chromosomes in the gametes produced by the F1 generation only have the big R allele. So, all males in the F2 generation would inherit the dominant red allele. There is no recessive Y allele on the Y chromosome. The recessive allele only exists on the X chromosomes. So, males in the F2 generation would never inherit recessive Y allele. No males can be white. E talks about another cross involving a red male and a white female. The question wants you to suggest an explanation for the result which is different from the prediction. There are two possibilities. First, it might be due to a mutation of the gene that codes for the body color. Mutation is a random change in the nucleotide sequence in DNA. This causes a change in the mRNA codon. A different amino acid is added to the polypeptide. When a different polypeptide or protein is produced, the phenotype would change. Another reason is crossing over. Crossing over is the exchange of genetic material between the non-sister chromatids of the homologous chromosome. The red male with an X small r and a Y big R chromosome experience this. The big R allele from a Y chromosome swapped to an X chromosome, resulting in a gamete with X big R and another with Y small r. When these gametes fuse with the female gametes, we will get X big R, X small r, which is a red female, and X small r, Y small r, which is a white male. 6a wants you to label structures A, B, and C. A is the cell that makes up the capillary wall. It is an endothelial cell. B is the physical barrier between the blood vessel and the Bowman's capsule. It is the basement membrane. C is podocyte, the cell that makes up Bowman's capsule's inner wall. B. Describe the role of structure B in ultrafiltration. The basement membrane is a physical barrier. It acts as the filter, determining the substances that can and cannot pass through. Anything with a relative molecular mass of over 68,000 to 70,000 is not allowed to pass through. Only substances that are smaller than this can become part of the glomerular filtrate. This stops large plasma proteins and red blood cells from moving out from the glomerulus as they are required to function in the blood. C wants you to calculate the percentage of water in the glomerular filtrate that is reabsorbed as the glomerular filtrate passes along the nephron. 180 dm³ of water forms part of the glomerular filtrate, but only 1.4 dm³ leaves the body in urine. That means 178.6 dm³ of water is reabsorbed. Divide this by the total and multiplying 100 will give you the percentage. Remember to give your answer to one decimal place. D. Describe how the action of ADH on the cells of the collecting duct leads to the reabsorption of water in the glomerular filtrate. All hormones are cell signaling molecules, so ADH is a cell signaling molecule too. Hormones affect their target cells by binding to the specific receptor on the cell surface membrane or inside the cell. The specific receptor is on the cell surface membrane of collecting duct cells. This is because ADH is a protein hormone and is hydrophilic. It cannot pass through the phospholipid bilayer, so the receptor cannot be located inside the cell. The binding stimulates the second messenger pathway. A second messenger called CAMP is produced inside the cell. This leads to an enzyme cascade, and the activation of kinase occurs. Enzyme cascade refers to a sequence of successive activation reactions involving enzymes. Then, vesicles containing aquaporins are phosphorylated. This is to activate the vesicles. They move towards the cell surface membrane and fuse with it. Aquaporins are added to the cell surface membrane. Aquaporins are membrane proteins that facilitate the movement of water across membranes. Their addition increases the membrane's permeability to water. More water moves out of the collecting duct by osmosis. It will then enter the collecting duct cells before moving into the tissue fluid and blood. 7a. Outline the sequence of events occurring after stimulation of the sarcoplasmic reticulum that results in the shortening of a sarcomere. When the sarcoplasmic reticulum is depolarized, voltage-gated calcium ion channels open. This causes calcium ions to leave the sarcoplasmic reticulum. They move down the concentration gradient. Calcium ions will bind to troponin. The binding causes troponin to change shape and tropomyosin to move. This exposes the binding site on actin. Note that this is not called an active site. Myosin head binds to the binding site, forming cross bridges. All the points here can give you 3 marks maximum. You must have at least 1 point from the subsequent events to get full marks. Then, myosin head tilts. Remember that the tilting is due to the release of ADP and inorganic phosphate from the head. It is not due to the hydrolysis of ATP. When the head tilts, it pulls actin. This is called a power stroke, and the sarcomere shortens as the thin filaments are pulled towards the M line. In B, we have a comparison of the diameter of striated muscle fibers taken from young mice and adult mice. You are asked to describe 2 differences between the muscle fibers of the mice and suggest how these differences may affect the sliding filament model. Young mice generally have more muscle fibers that are smaller in diameter. Their graph shows a smaller range of diameter. Quoting in comparative data will give you a mark. For example, you can quote in the range for both groups to support point number 2. With a smaller diameter, the young mice would have fewer muscle fibers or sarcomeres. This means there will be less actin and myosin, the protein that makes up muscle fiber. This causes fewer actin-myosin interactions, leading to a weaker contraction. 8a shows a diagram of chloroplast. K is the colorless fluid surrounding the grana within the chloroplast, stroma. L is the thylakoid membrane, an internal system of interconnected membranes. M is the space enclosed by the thylakoid membrane. It is called thylakoid space or lumen. N is a stack of thylakoid. It is called a granum. There will be a high concentration of protons in M, the thylakoid lumen. This is because the ETC on the thylakoid membrane moves protons from the stroma to the lumen. Photosynthetic pigments are found in the photosystem embedded in the thylakoid membrane. The granum is an acceptable answer as well. Light-independent stage occurs in the stroma, as that is where the Kelvin cycle occurs. Light-independent stage occurs on the thylakoid membrane or granum as it involves photosystem. B. Describe the role of other photosynthetic pigments found in plant chloroplasts. Other photosynthetic pigments that are not primary pigments are called accessory pigments. They form light harvesting complexes called the antenna complex. Giving one example can get you a mark too. The accessory pigments absorb light energy or photons. However, they cannot emit electrons. They pass the energy on to chlorophyll A at the reaction center. Different pigments absorb different wavelengths of light. They can absorb the wavelengths that are not absorbed by chlorophyll A. They maximize the wavelengths that can be absorbed, leading to a higher rate of photosynthesis. In C, we have a graph that shows the absorbance of the whole chloroplast and the pigment extract. The question wants you to describe the differences between the two spectra and suggest explanations for the differences. The absorption is higher for whole chloroplast throughout. You can use comparative data to get a second mark. For example, you can compare their absorbance at 500 nanometer. You can also get this mark by stating the wavelength which gives the greatest difference. It is at 530 nanometer. The whole chloroplast has a higher absorbance as the pigments are arranged in the photosystem. They are held in a better position to have a greater absorption of photons. Besides, whole chloroplasts contain more pigments. Some of the pigments may be destroyed or lost in the preparation of the extract. In 9a, we have a diagram of a synapse that uses dopamine as the neurotransmitter. 1. Describe how the release of dopamine from the presynaptic neuron can lead to an action potential in the postsynaptic neuron. The question mentioned that its action is similar to that of acetylcholine. So, just use your knowledge about the cholinergic synapse to answer this question. After being released, dopamine diffuses across the synaptic cleft. It moves down the concentration gradient. Then, it binds to the specific receptor. The receptors are found on the postsynaptic membrane. The shape of dopamine is complementary to the binding site of the receptors. The binding causes the ligand-gated sodium ion channels to open. This leads to an influx of sodium ion into the postsynaptic neuron. Since sodium ion is positively charged, the depolarization of the postsynaptic membrane occurs. The inside of the membrane becomes more positive. The action potential is created when the threshold is exceeded. Another compound in the body produced from dopa is dopaquinone. It can be converted into melanin, so you can also use melanin as the answer. We learned this in the inheritance topic. People without albinism can convert tyrosine to dopa, dopaquinone, and melanin. Melanin is the pigment responsible for our eye, hair, and skin color. B. Suggest and explain whether an action potential would be generated in the postsynaptic neuron if GABA is released into a brain synapse. Chloride ions are negatively charged, so their influx following the binding of GABA causes the inside of the postsynaptic neuron to be more negative. This is hyperpolarization, where the inside of the neuron is more negative than the resting potential. More sodium ion is now required to reverse the membrane potential. When there is not enough sodium ion entering the neuron, it is less likely to reach the threshold. No depolarization of the postsynaptic membrane occurs since it does not become more positive inside. So, no action potential will be created. Question 10 is about the moose. A. Describe the main differences between the kingdom Animalia and the kingdom Plantae. In this type of question, remember to make side-by-side comparisons. Do not write a whole paragraph about Animalia and then another about Plantae. First, you can compare the presence of cell walls, permanent vacuole, and chloroplasts. The feeding mood is not the same. Animalia is a heterotroph because we cannot produce our organic compound. Plantae is an autotroph as they can carry out photosynthesis. The storage carbohydrate is glycogen for Animalia and starch for Plantae. Note that animals can't synthesize starch from glucose. Only animals have a nervous system for control and coordination. Most members in the Animalia kingdom show movement from place to place, but Plantae does not. In B, we have a graph that shows the temperature difference of each decade compared to the 20th century. 1 wants you to calculate the rate of increase in temperature per decade between 1980 and 2020. First, find the difference between the starting and the end point. Then, divide by 4 as they are 4 decades from 1980 to 2020. 2. Suggest and explain reasons for the decrease in moose population. Climate change may cause the temperature to exceed the range that can be tolerated by moose. A reduction of food can cause increased competition between the organisms. Climate change may also lead to a decrease in snow-covered areas. Predation and hunting can become easier when the snow depth decreases. They may have lost their habitat due to human activities such as deforestation. Lastly, new disease can be a reason for their death as well. Infections may be introduced by invasive species. It can also be related to an increased ambient temperature. That's all for today. If you think my videos are useful, don't forget to like, comment and subscribe. Thank you for watching and see you again soon.